Hey, this is Dr. Ben Morrill. Welcome to episode 17 of Reptile Genetics Weekly. We've got a fun one for you today. Some cool things to talk about. It's been very busy, very crazy. Been working hard and keeping Shawn and Kayla very busy as well. But it'll be really fun to be able to talk about all these things we've got going. And we got a whole bunch of results out today. The majority of them, we've still got some more that will come out tonight and then tomorrow. The last ones that are going out are some of the genes for the panels, but most things are out. The run did go pretty well. Overall, things worked well. The only thing we have are some of the genes for the panels did fail. So we'll start another run here pretty quick. So we will probably have the next batch of results about the end of next week. So about a week and a half away. So that's where we're sitting as far as results. And got both Shawn and Kayla here. How are y'all doing?
Hey, we're doing good, Ben. It's good to see you. Hello. And hello to everybody who's in the stream. You guys were actually ready before we were. We got Kate Rose, Leviathan Snakes, Prophecy Exotics. I can speak, I promise. Nixon Reptiles, whole bunch of folks. Hi guys, it's great to see you.
Yeah, so we've got a bunch on Instagram as well. So, and any of you on Instagram, if you want to see the pictures and things, jump onto Facebook or YouTube. But otherwise, you can just listen and hear the audio and see me if you look at your picture.
Yeah.
All righty. So we do also have a live results to do. We've got Courtney and Steven from Leviathan Snakes here with us.
Hey guys, welcome.
So how long have you been waiting for these results?
Well, all right. So for the test, it's been like what? You put out the test, 2 weeks ago, 3 weeks ago? Like something like that. It's been pretty quick.
Yeah,
but
3,
in general, since we got the babies, like 2 years.
They're both females?
Yeah. No, they're both females. So we picked them up. It was 2 years ago at Daytona, I think. Yeah. Maybe a little bit more than 2 years ago. But we had gotten a leopard sunset female from Brad Boa and Brock Wagner, and she was amazing, incredible animal. And they threw in these two girls.
About a lot of snakes.
Yeah. They threw in these two girls with that. So we're like, oh, we're just starting out in the Sunset Project. We were super, super excited. And now the fact that we get to know before they have a clutch, even though we're pairing them. So
yeah.
Yeah.
Love that. So
all right. So tell us about Viola.
Viola's the leopard. I think that one is Hazel.
Hazel.
That's the actually normal head
here we go. There's Viola.
Rude. She's rude.
She's probably the meanest snake we have, to be honest with you. When I had to get we took pictures of her today, and I felt like I was handling like a feisty boa. She doesn't back down.
Yeah, she's not scared. She'll take your lunch money.
Yeah.
I love her.
Yeah, so she's actually being paired to our visual sunset right now because she's up to size, and we were gonna prove her out either way.
Yeah.
Well, now you'll find out if she's worth it, huh?
Yeah. Now we get to find out. So
yeah.
All right. So Hazel's the other one.
Yep. Hazel's the normal.
Do you have that picture? Yeah, there you go.
She's a bit smaller. She's really bright for a normal, but she's a lot nicer than her sister, but she doesn't eat quite as well.
Yeah. Her sister, she'll eat anything. So
including me.
Yeah.
All righty. Well, I guess now is the time.
Yeah.
Right.
Okay.
Which
So
one do you want to hear first?
Yeah.
Let's do Hazel first.
Hazel.
So that's Viola.
Oh,
right.
There we go. Sorry.
Yeah, there we go. Hazel.
So Hazel is not ahead.
Dang. All right.
So okay. It saves us some time.
Yeah, saves us some time.
Saves us a whole clutch.
Yeah.
But your mean one is worth it.
Oh boy, I'm excited. Oh, that's awesome. That's super freaking cool.
Okay.
If you're gonna get one of them, might as well be the leopard.
We were like, he wouldn't ask us to be on a live if they were both negative, would he?
No.
Just crush our dreams in front of everybody.
I am a scientist. so I'm not the best at this, but I do know at least not to have you come on for all that.
Yeah.
Oh, that's awesome.
This is so cool. Now we know that we're expecting another visual sunset, a leopard sunset collection.
Yeah,
this
that's
year.
awesome.
That is,
I'm super freaking excited.
that's
Thank you
exciting.
guys so much. Like the work that you guys have done, not just with the sunset gene, but with everything, 100% has completely changed the ball python breeding hobby.
Yeah.
Like it's really, really cool. And we were planning out our pairings today, and we have a clown 100% het sunset male. And because of the stuff that you guys are doing, we are like, oh, let's pair him to our super asphalt rather than pairing a visual sunset, because we can go for the super asphalt sunset clown right away, even though we only have a clown 100% het sunset.
Because we don't worry about the 50%, we can just test all those sheds now.
Exactly. It
Yeah.
completely changes. It makes it opens up so many more opportunities. It's just awesome. The stuff you guys have done has completely changed the hobby, and we really, really appreciate it.
Well, I appreciate you all sending sheds in. That's originally where we started interacting. You guys jumped on and got sheds to me to help when I was designing the sunset, and they got used, and that was very helpful to get to where we could do this.
No, we're super, super excited. So, and Brandon, he was messaging us. He wants to do this with you guys too. So I don't know if he's reached out to you guys yet, but you guys got to bring him on. Get the results.
All right.
The live stuff is awesome.
Yeah, and I apologize to anyone. These last couple of weeks have been crazy. I think there were 2 or 3 people that did have results in this run, and I might have missed, being able to save them back. But we'll do a better job. So message one of us and let us know order number and shed ID or IDs, and I'll make sure I do a better job. But yeah, this last couple of weeks have been crazy.
Awesome.
And I was going to say, I really enjoy your videos talking about philosophy and talking about planning and, kind of the strategy that you're going to use. I enjoy, and your production quality is infinitely better than mine. That's for sure.
We got to
Really nice production quality.
I know we've talked about it, but we got to jump up there sometime, and we got to do like an in-person collab. It'll be awesome. So
that would be cool.
Yeah.
So.
That sounds good. Well, thanks for coming on.
Yeah.
Awesome. Thank you guys.
Thank you guys. See
Bye.
ya.
Yeah.
All right. So what do we got next, Ben?
All right. Well, the other really cool exciting thing we've got going on is we are very, very close on 3 more tests. So I'll say right now, don't start asking Clutch or Morph Market for these yet. I still need to do a little bit more work, but they are to the point where I'm pretty confident that we can talk about them, and it'll still be a couple, 2 or 3 more weeks before I have it polished off and fully verified, and we have it available through our vendors. But the big one that I've been working on, and the first set of 24 look like they all match that were known samples, is for Redstripe. And so I'm pretty excited about that. There's a lot of people that have asked about Redstripe, and so it looks like we'll have that test here pretty quick, and I think that'll be pretty helpful for people.
Mm-hmm.
And then talking back and forth with Will from Royal Canadian Reptiles, he's talking with me about trying to figure out the best way to get testing done in Canada, and I ended up talking with the group in Canada, Alan and Heather. I've been talking specifically to Heather. Those of you that have watched our videos, I think it was our 2nd episode where we talked about the pied anole lizards. So they were the ones that found piebald mutation in ball pythons, and then they worked together with another group here in the United States in Georgia to do the piebald anole lizard. Well, so I've been talking with them, and they have found VPI Axanthic and Cryptic.
Yeah,
so,
VPI Axanthic and Cryptic.
Yes, VPI Axanthic and Cryptic. And so after a little bit of negotiations back and forth, we decided to make a trade. And so I gave them some information on being able to do tests for genetic stripe, Enchi, and black pastel. And Heather's very experienced in being able to sequence specific genes and be able to find, essentially the other members of that black pastel complex or 8-ball complex. So Heather's going to start working on that, and we'll certainly update y'all and let you know as she gets, cinnamon some of those other het rate exanthic, some of those other mutations that are in that same, either that same gene or close by. So she'll start working on those. So yeah, so those of you that are listening in Canada, you'll have a few more tests that Heather can help you with. And then those of you in the United States, I just need to validate that I can get these tests to work in my hands. They're working for Heather, and she'll be doing the same thing, making sure my tests work for her in Canada. But as soon as those look good in my hands, then we will make them available.
All right. So excited to be able to announce this to you guys. I love that somebody in the chat was talking about, like fingers crossed, please be VPI Axanthic. So as soon as I saw that, I was like, oh yeah, man.
I think that's one of the ones what would you say, Shawn? Is that one of the top 3, if not the top one, that we have people ask questions at least weekly, if not daily?
Interfacing with customers mostly every day. Yeah. VPI Axanthic's definitely top 3. Yeah. TSK is pretty close, but VPI is probably top 3 for sure.
Mm-hmm.
Yep. And in Canada, Heather and Alan are working on MJ Axanthic, and then we've got a bunch of shed sequence information coming in the next couple of weeks for TSK. So there will be more Axanthic information and tests coming soon.
Sweet. That'll be great to see. And of course, you guys will know as basically as soon as we know, as soon as that goes live on Morph Market and Clutch. So that'll be really good. And you said only a few weeks away, right, Ben? More than likely.
Yeah. They're all 3 at a point where it should not take very much more. Probably just need to be on the next run. I'll do some, another 50 to 100 and make sure that they're known samples. Make sure they work like we expect them to. What'd you say, Shawn?
I said I always say it's the test.
Yes.
Test the test. Yep.
This is the very last part where we just make sure we test everything as much as we can with all the sheds that we've got to make sure that the results match what we suspect they should be. So.
Yeah, and we've mentioned it before, but essentially what we're going for is at least running 100 and have at least 99%, over 99% accuracy. So if we run 100 and they all work like we expect, and we're putting about a third of them being homozygous, a third being hets, and a third being non, we know they don't have the gene. And as long as that 100 all matches what the expected was, then we go ahead and make that test available. So any of the tests we have available, you can expect them to be over 99% accurate. And every month that goes by, that accuracy will get higher and higher. And if we find a problem, then we'll redesign and fix it. So we haven't had to do that yet, but it might happen on one of these.
All right. Well, good stuff. So next, I think we're planning on doing like a little Q&A for the next little while, right?
Yeah.
Sweet.
Do you see that Nixon Reptiles question? That's a good one to hit right now.
I saw that one too.
Yeah. Let me see. If one of you guys find it before I do.
Third from the bottom.
All right.
From the bottom. There we go.
So.
Yeah. How's it looking for the other line of Ultramel?
So it's looking good. I want to say Heather, because we're just talking about Heather. Hannah at Eastern, Dr. Seidel at Eastern Michigan University. She has something that looks like is working. And I actually haven't replied to her email. I need to do that. But she's given me some information that I will be testing out and make sure that I can get that to work in my hands. But it's looking good, looking like we'll be able to cover both of the Ultramel both the mutations that cause Ultramel. So far, we've been able to do the first one that Dr. Seidel published, and now it looks like we'll be able to do both. So that's another one that within the next couple of weeks, we should be able to do that as well.
Good stuff. All right, and we have from Home Run Balls, any updates on Puzzle?
So Puzzle is in the same spot as TSK Axanthic. We are just waiting to get results from the big sequencing run that we did on the big expensive machine. So we'll have a pile of data for TSK and for Puzzle and for Monarch. All of those will be coming. It's supposed to be in the next couple of weeks. Hopefully they'll get it to us here soon. And we have enough samples in those runs that it should be fairly quick, unless we hit something crazy like Desert Ghost. That's been the weirdest one. Unfortunately, that was the second one we were trying to make work, and we spent forever on it. But things are moving along faster now. So yeah, hopefully here pretty soon.
Good stuff. And we have from Exotics, any word on Monsoon?
So since talking about that on here and lots of people asking, I've had a lot of sheds come in. So that's awesome. I should be able to send those out. We've been kind of waiting till we get the data back for Monarch, TSK, and Puzzle. But once we have that, I'll send out, or maybe even before if Shawn tells me to just get it done. But we'll send in Monsoon sheds. And so since those still have to go to sequencing, that's probably like 3 months out at least, because we have to get them sequenced and then do the analysis, then design the test, then validate it. So
okay, looks like we lost Shawn, but he'll probably come back. He usually doesn't go for long. Let's see. We have from Elite Exotics Ball Pythons. John, are you guys going to Pomona?
I am. I'll be the lone representative at Pomona. I'll be at Dark Cloud Reptiles table. He found a way to give me a little space, and unfortunately Shawn's not on here. He could give us a little bit more detail about what I'll be able to bring. We've got some new designs that we're working on that will only be available at the shows, and it'll just depend on whether they can get done in time. Otherwise, maybe I'll just have some example ones and take orders. But yeah, I will definitely be there. You'll see the hat, you'll see the beard. Come say hi.
Yeah, seriously, like Shawn actually has been working on getting some really nice. Welcome back, by the way, Shawn. Has been working on getting really nice designs going for our merch. So it's not just the merch that's in the store. This will be show specific stuff. So if you go to a show, we'll be there, or you see our logo somewhere, run up. There's probably going to be some really cool merch, and like I've been getting, the design mock-ups that Shawn's been sending in, and they're really cool.
So collectively, we all sort of came up with the idea that we'd like to come up with every year, maybe 1 or 2 new designs that RGI comes out with, representing the new tests that we created in that year, but also just neat artwork to put on a hat or on a shirt or whatever. So we've got 2 shirt designs and 1 design that's going to go on a hat. These are going to be show only. So either we have them at the shows, or you order them from us at the shows and we ship them to you because we don't have them yet. But they won't go on the merch store probably in the first year. If we do put them on the merch store in the future, it will have something else on it that sort of shows that it's not the collector's edition. Let's put it that way. It'll have something else on it other than, the standard artwork. So anyway, it's just kind of a neat idea. I'm one of those nerds that likes to collect stuff like that. And, we're a brand new company, but everybody gets to kind of take this ride with us, as you saw in the live reveal earlier.
Yep.
And I just think it's kind of neat if, we have something else that we can toss out to you guys that, hey, I've been with you guys since the beginning, and here's all 10 shirts, hanging up in my closet, and that'd be cool. I'd like to see that.
Same here. We have from HB Reptiles, is lace in the works?
I don't have enough sheds of lace yet. I have a select few, but not enough yet. So if you've got lace, definitely send it in.
Yeah, because you said we needed at least roughly 20 good sheds to even begin to have something that would work for each thing. So, and ideally you're going to have as much genetic diversity between those as well. So if they're all from the same male and 2 females, it's harder there.
Yeah.
Okay.
Yep. Yeah, 20 is where we may start, but it might not work. But it could. Having 40 or 50 is what I would prefer.
Yeah. Let's see. We have from Hades Herps. Can't wait to see. Oh, I thought that was a question. There was a question near that. Anyway, hi Hades Herps.
If it was in this last run, then I think Shawn, did you do all the DG updates yet, Shawn? Or are you still in the middle of
everything that was on the first spreadsheets is done. So if the DG was run, it's up there. There's I think a couple of DG still pending, but they're going to be on this run that's coming up this week.
That's right. That's why I highlighted it, because I wanted to get an update from about the DG.
There are some people that just came in a little late on sending a shed in. It was on the last run, but then added DG to the order. And it's like, I'm going to have to resubmit it now. It's one of those sheds that we talked about where I have to pull it again and stuff it again and send it off because it just didn't get here in time. So there's a few of those out there, but
yeah,
everybody else that ordered regularly and normally, all that stuff was updated today. So
sweet. You don't have that box that you showed me earlier, like right before we started streaming, where it was just like full of sheds and
you want me to get it?
Yeah, I think they'd love to see that.
This
is Shawn's life.
Information.
This is my life every day.
Thank you all. We asked for it. You gave it to us.
Yes,
it's baby season. We asked for it. That's right. So that's
exactly, and we love it.
expected.
Awesome.
As kind of an aside, is everybody that's watching, just because your order is open doesn't mean there's not results in there. I've explained multiple times to people every time we update results, particularly with panels. We don't just get all of the results all at one time. It comes to us in a big mass of data, and Ben's got to sort through that and translate the data into results. As those are translated, he sends them to me and I update them into the system. So you can have, say, a panel open for 2 or 3 days. And if you check it every few hours or once a day, you'll notice that more and more things are being updated to it. So just because you haven't gotten an email that says your order's not completed, doesn't mean that there aren't results in there, specifically maybe the ones you're looking for. So check early because there may be results in there.
Yeah.
Or if we have a bad run, if we have a panel run where something goes bad and we have to delay that one result for a week, I'll still have all the other 15 genes updated waiting on that one. So I can't close it until it's done, but you can get your results.
Yeah. Sorry if you guys heard something outside there. We have a road where lots of people like to zoom past loudly. Cool. And this is related to Shawn's question, I think, regarding this. Where can someone find the spreadsheet you were referring to?
What spreadsheet? Me?
Yeah. One of those.
I think that you were talking about. I asked if you had results up, so the spreadsheet Shawn was talking about with that's
between us.
Oh, okay, that's internal stuff. Got it.
When I go through the hundreds of calls for the tests, then I send that to Shawn, and he's the one that actually enters it into Morph Market and
Yeah. So. Let me just explain the spreadsheet. This last run was just shy of 700 samples that we did. Probably a third of those were panels with anywhere from 12 to 16 tests on each of those orders.
Wow.
So do the math. It's a lot of translation work for Ben and a lot of updating for me when I have to go in and update 16 genes for a particular panel. It takes a minute. So we have a spreadsheet where we are both actively marking and adding to and taking away from at all times so that, as soon as he sees an update, I can see it and I can update it into the system.
Good to know.
Yes, you don't want to see that spreadsheet.
Yeah, you really don't.
I've seen glimpses of it. It's a lot.
Well, we can talk a little bit about the process. I mean, it starts here where we receive the sheds in the system, but then as we stuff them into strip tubes for testing, they're added to the spreadsheet. And then Ben uses the spreadsheet to further prep them and get them ready for sequencing. And when they come back from sequencing and he translates results, then he adds results to the spreadsheet. And then I pull the spreadsheet, print it, and update them back into Morph Market. So it's a bunch of different steps for you to actually get your results. It takes a minute.
All right. So another question from EH Exotics. 100% het sheds, are they helpful for Monsoon or anything? Any other morph you're submitting?
Yeah. Any of them we don't already have. Het sheds are helpful. The homozygous ones are the best because we have twice as much information to tell us what that morph is. But yeah, the het, like right now, if you could send HepMonsoon, 100% HepMonsoon sheds within the next week or 2, 3 at the most, then they will probably make it on this big run that I'll do for Monsoon. So that would be right now is the opportune time to send Monsoon sheds in.
All right. Good
and
to know.
Make sure you go to our help us on our website, and that will make sure that you're sending, if it's for a new test, you want it to go to the Virginia PO box. So that'll come straight to me. And when I prep them to go for the big sequencing run, then I'll be able to do that a little faster because it'll come straight to me.
Yep. And I just played the sequence there on there. How you get there? Yeah, raregeneticsInc.com/help. That's all it is. And we also post that in all of our, like, the descriptions of all of our videos. So after this live stream, it'll be updated there too. So, yeah. Let's see. We have from Ted Stanhope. Doc, have you been getting any sheds in for GCR Axanthic line?
I don't believe I have. So if you or someone you know has a few, definitely send them in. This is a good time. Not only, we touched on this briefly on one of the other lives, but not only in ball pythons are we working on Axanthic, but there's some colubrids and other python species that we're working on Axanthic. So this is a good time to send that in. We're thinking about Axanthic a lot.
All right. Let's see. We have also from Hades Herps, with Desert Ghost being polygenic, do we still call it het?
I think that most people have been. I don't know for sure how it'll be over time. For me personally, I'll talk about the breeders that I have, and I actually just hatched out a clutch that was from a clown het DG male. So those babies I'm going to test, and when I do, if I sell any of them, I'll just put up, whether they're het for DGA and or DGB. I know that the het male is het for both DGA and DGB. So I'll have some babies that'll be het for both, some babies that are het for one or the other, and then some babies that don't get either. So yeah, I think that if you wanted to say it in more of like a genetics way, you would call it a non-visual carrier. And so a non-visual carrier, if you're talking about a single gene, that's what a het is for a recessive trait. It's a non-visual carrier of that genetic trait. And so if it's het for DGA or if it's het for DGB, either one, it's going to be a non-visual carrier. And so that's exactly the same kind of a thing. It's just there's potentially two different places that it could be het. So I think, if it's het for A or for B or for both, it makes sense to still call it a het. But some people may want to make sure that you're letting them know specifically if it's het for A or B or both. But it kind of depends on you all, how specific you want to be. For me, as a genetics nerd and a snake breeder nerd, I definitely want to know all that information and take that into account when I'm making pairings.
Yeah.
From the business side, the software guys, we talk about this too. I mean, there's really not a good way, because this polygenic trait works very similar to a het trait doesn't mean they all do. And we may encounter some genes in the future where something's polygenic and calling it het really doesn't make a lot of sense. And we're trying to figure out the best way to navigate that. Right now, we're just still using the terms homozygous and het as far as results go on both software platforms, just because we don't really have a better way to, through the software, let you guys know that. Sooner or later, I think we may run into a gene where that becomes a problem though. So it's just something to think about. Using the proper terminology, as lay people is pretty important, even if right now, software wise, we've got to use some old technology just to get the information across, or some old terminology rather, just to get the information across.
Yeah. So, I mean, it'll be some kind of a joint venture between the hobby and where the hobby goes, and also what can be easily translated from the genetic side too. Yeah. I'm interested to see where that'll go too. Let's see. We have from Elite Exotics Ball Pythons. Would you like some zebra sheds and het zebra sheds?
Yes. Yeah. I have very few of those. I think less than 5. So yeah, that would be excellent.
Yeah. Go find all your zebra fans and send them on in. Sweet. This is from Bod's Exotic Reptiles. Would I be able to send in the very first shed from a hatchling, or is it better to wait 2 or 3 sheds?
So the really quick answer to that is it depends on how you are keeping your hatchlings right out of the egg. So if you separate them right out of the egg, then that first shed could be fine as long as you're not keeping them super wet all the time. So I know for me, hatching out ball pythons, and when I hatched out, baby boas as well, I kept the whole clutch or litter together, and I kept them really humid and moist until their first sheds. So if you do that, then that's not going to work, because we found out from garter snakes, if it's a wet environment and there's animals climbing on each other, there can be some cross contamination.
Oh yeah.
So you do want to, if you want to be able to use the first shed, then as soon as they hatch out, go ahead and set them up individually, and you can keep them fairly wet until their eyes, through when they go blue, just when they clear up. So, it's within a day or two that they're going to shed. That's when you'd want to make sure it's not like super wet in the cage. You don't want wet paper towel all day for that shed to get placed onto, because that moisture is what will destroy the DNA. So if you separate them right away, and then you watch, and when their eyes are clearing up and they're about to shed, you kind of decrease, make sure it's dry in there for when they actually shed. Then the first sheds work great.
All right. Oh, here's a cool. Here, this is from Hades Herps. Do y'all have Hurricane down yet?
A few sheds have come in because we talked about Hurricane. I think I had about 3 or 4 or 5, and I think another 3 or 4 or 5 have come in. But I definitely need more.
I've got about a dozen that were sent here from a customer sent a bunch of Hurricane stuff in along with their paid samples. Perfect. So my next shipment to Ben will include probably another dozen or so Hurricanes.
Oh, nice.
Cool.
Good. Yeah, send them on in if you have them.
Yes.
And this is a question from Leviathan Snakes. Quick question: would an egg contain the genetic info from the mom or the baby? I guess like the amniotic fluid or a scraping from the egg. I'm not sure if that would be plausible.
Yeah. So one thing that they do with, first of all, to answer whether it's from the mom or the baby. So right after the egg is laid, if you were to swab the outside of the egg, you'd get mom's DNA. But once it's sat there in an incubator, if it was with the mom, maybe the mom's DNA stays on it. But if you're doing artificial incubation, by the time those eggs hatch, mom's DNA is long gone because of the high humidity. And then what's on the inside of the egg is all baby DNA. So in birds, for bird sex determination, for years, they have been using like a scraping from the inside of an egg. And so you could, if you wanted to separate even earlier, you could separate eggs, and maybe once they're mostly have absorbed and they're close to coming out of the egg, you could put them in separate cages, and then that way you could just take a piece of the inside. So you'd want some of the membrane, so it will go from being vasculated, having those red veins, and then it'll get to where it's clear, but there will still be some membrane there, and it will dry out and get a little crusty. So it might be more like a scraping, but yeah, that should work just fine. I haven't personally tried it, but it should work. And if you really wanted to get crazy, even if you didn't cut your eggs, if you wanted to, when they're close to hatching, you could actually use a needle and go into a vein and get some blood and just put a blood spot on a piece of paper. And I know blood spots work really well. But waiting till they hatch out and shed, usually you got to wait till they're feeding anyway. So I don't know if it's worth messing with that, but any of those should be, potential ways to get DNA.
Good stuff. All right. We have, from Mutant Geckos. Do you want blue, brown, and green Barons racer sheds?
If there is something that keepers thought that would be helpful for, we certainly could work down that path. The only thing we're doing right now with racers is sex determination. Mm-hmm. So if there is a question for coloration or for the locality or anything like that, we would need a bunch of sheds. Whatever kind of a test you want to make, we would need a pretty good amount of sheds that we know are either different colors or different localities or whatever. And then we could design something. So I guess that's kind of a question for the Baron racer keepers. If that's a test that would be helpful, and get sheds together and send them in, and we will go from there.
Yeah. All right. We have Actually, do we have anything on Instagram right now while I'm looking through the comments?
Let's see. Yeah. So here's Royally Morphidelic says, if we suspect Axanthic but not sure of VPI, TSK, etc., how would you prefer we request specifically what to test for? So for right now, in a couple of weeks when we have VPI Axanthic available, that will be the only choice. So all you could do is use that as a way to eliminate. So if you got a negative, then you wouldn't know for sure what line of Axanthic it is. You just know it's not VPI. But if you get a positive, then you know it's VPI. In the future, when we have TSK and maybe the researchers in Canada get MJ and we get some more sheds for some of the others, then you just have to decide if you want to pay for one test or all of them, or however you want to do that. But we will definitely have that marked so that you know which of the Axanthic lines that are being tested for.
All right. What else do we have on Instagram? If you can spot any.
There's another Ultramel question. So we did talk about that earlier. We are very close. Within the next couple of weeks, we should be able to test for both. Dr. Hannah Siddell has worked on that and has something that looks like it's working. So we're about there.
Yeah, Dr. Seidel.
Yes, thank goodness. Purple Orchid Exotics says, I have a normal piebald pos het albino. Can I send his shed in to test? Yes. So the test you would want to choose, you can go to either Morph Market or to Clutch in their genetic testing section and just choose to pay for the het albino test and send the shed in. They'll give you an email that will show you what to write on the bag as you send that in. But yeah, that test works really well. And that's another one that Dr. Siddell and her students at Eastern Michigan University that they figured out, and it works good.
Good to hear. Let's see. This one isn't a question, just a comment that I like from Mike Williamson. Pins Pythons here. Chocolate and Garcia chocolate on the way. Shed tests or sheds for submission. They love their RGI shirt and feel smarter when I put it on, which is a big leap.
Right on.
So, awesome. Love to hear that.
Yes.
Please feel free to post a selfie in it and tag us or send it in a message or something. We'd love to see that. So
I did get my Pins Python shirt as well. So I'll have to start wearing some of those. So put the camera back a little further and start wearing some shirts that y'all send in. Yeah. I did get one from you. So I'll wear that one of these days.
Yeah, that would be good. I'll see if I can remind you next week.
There you go.
Yeah. We have from Adrian Ramos. Do we have Hognose morphs in line?
Yeah, so we have started getting sheds. I would say for other species, and Shawn, feel free to jump in from what you're seeing. I'm seeing a lot more corn snake than anything else, but definitely some hogs coming in as well.
Yeah, there's a lot of corn morphs coming in. We've talked about this many times, but we keep any of the sheds that have mutations that we would be interested in developing a test, as long as you list them on either the Clutch or Morph Market sites when you are purchasing a test. If I can see that it has something in it that we can use in a future test, we save it. So please use those fields, fill them out with the mutations that you know that your animal is either homozygous or heterozygous for.
Yeah. All right, and from Scales and Tails Breeding, we have, I've only ever had one shed that needed to be rerun. Haven't gotten a new shed yet, but what would cause a rerun, or what can cause a rerun?
So great question. The number one problem is that shed got wet or stayed wet too long. Every once in a while, I'll get a shed here and it's like, I can see in the bag, it's wet in the bag.
Oh, no.
So it got sealed wet. And those ones just go directly in the trash. If the shed stays wet for very long, it's going to destroy the DNA. That water just tears the DNA up. The DNA is a pretty stable molecule, but the biggest enemy is water. So more than likely what happened is that shed sat in the cage just a little too long. So the best thing you can do, like when I have sheds I'm watching for, I check every morning and every night. And so at the very most it could be sitting there is, 8, maybe 10 hours, something like that. and I pull the shed out. I flatten it out as much as I can. I look for the driest and cleanest, flattest part of it, and just take that part, unless it's like a really small snake, like maybe if you're trying to get a really small colubrid baby for sex determination, then maybe you need to send the whole shed in. But like for a baby ball python, we just need a piece size of a quarter. That's plenty. So like if you do like an inch long of, a cross section of the baby ball python shed, that's enough to do like 50 tests on.
The other thing folks should know is that by the time you see a rerun result in the system, we've already tested it. It came back as a rerun. We repulled it, restuffed it, and tested it a second time. So if you get a rerun result, that means we've tried and failed twice to extract DNA from those cells, and we really need you to attempt to send another shed in, because every one of those times that we make an attempt has a cost attached to it. So we're willing to try twice before we push it back into your court. So when you see a rerun result, that means we've pulled two sheds from your sample, two samples from your shed rather, and run them through and failed to get DNA both times. So
that's good to know. I actually didn't even know that. So good stuff. We have from Michelle Spencer, vanilla versus fire tests.
So we are very close on fire. I don't have enough vanilla tests of vanilla sheds yet, but yeah, we're very close on fire. And I think either Dr. Sadell or Dr. Rafi, Heather or Hannah, I think that one of the two of them may be interested in working on that complex. So all of this takes time. When we talk about going after a complex, this is months, not days or weeks. But yeah, it's something in the works. But the first one in that black-eyed Lucy complex that we personally will have at RGI will be fire.
Sweet. And from Jesse Corte, if we have sheds that we want to do all of the tests for, so a panel, including the cryptic gene, do we need to wait until the cryptic gene is available, or can we send in now requesting to run that test as well later? So I guess doing the panel later down the line when cryptic is released? I'm not sure.
Yeah, you definitely want to wait until. So in this case, where we know we're really close, if cryptic's important for you, I would hold on to that shed for another couple weeks and make sure when you send it in, that's in the system. The choice few of you have had to go back and forth with Shawn and I. If something is just a little different, not in the system just right, sometimes it can end up being a back and forth for several weeks. If it goes through the system exactly how you want it, then your chances of getting your results in 2 to 3 weeks how you want them is much higher. So that's just a lot safer. If you really want to send it in how it is right now and try to work with us, you can try. But if you want to make sure it gets done right and in a timely manner and less stress to everyone, I would just wait. Especially for those three, I'd just wait a couple weeks and hopefully we'll have those available.
All right.
Can I say something real quick? So there is a kind of a unique thing that happens with both software platforms, and that is if you order a panel today, but don't actually send me a shed for 3 months, when I go to update results, it's only going to list the mutations that were on the panel today. So 3 or 4 months from now, if we've added a half a dozen new tests, I'll probably have results for those as part of the panel, but I won't have anywhere to put them because the software doesn't have that gene as listed in the panel when you purchased it. So in those cases, kind of the sideways answer to your question is, when you order a panel now, but you send a shed in 3 months from now, if you order today and you send me a shed 3 months from now and cryptic is part of the panel, you will get a result. It's just not going to be where you can easily see it. It's going to be in a notes section or in some other fashion. I'm going to have to email it to you or something. So there is this weird thing that happens when if you wait too long to send a shed on a panel, the number of tests that I can easily update is going to be smaller than the number of results that you have every single time. I encounter that with almost every panel that I do. Unless you purchase a panel today, send me a shed tomorrow, and I get results to you within 3 weeks, we're probably going to have another test or two added to it within a month. So there has to be a way for me to do that. So right now, there's a notes section on Morph Market, and then with Clutch, unfortunately, there's really not a place for me to do that. So I have to email everybody and go, hey, here's your results through the system, and here's the stuff that you hit that isn't in the system yet.
And yeah, one of them that I caught in this run was someone that had a white snake that was possibly het for clown or clown, and then also possibly het for genetic stripe or genetic stripe. And, they said in the notes, but I know genetic stripe isn't available right now, so they must have paid for it, a couple months ago.
Yeah,
it's going through. But it ended up being a G stripe. So.
I called a customer today that had a panel from months ago, so Enchi was not included, but it came out in our panel results, and they had no idea that Enchi was even in the animal, and it turned a 5 gene into a 6 gene animal. So those are the phone calls that I make, because those are the ones hey man, guess what?
Yeah.
So those are the fun ones, and that does happen sometimes. So people have no idea what's in there until they run the panel, and we don't hit them that often. And when we do, it's usually an additional gene, making it a 3 gene or maybe a 4 gene. When you hit the 5s and 6s, those are fun. And today was a 6 gene. The guy had no idea there was NG in it. When I called him, he was like, yeah.
I wish we'd gotten him on the live stream too.
He hit 2 pos hets, so he hit both hets. I forget what they were now. But then I said, also, is there any NG anywhere in there? And he goes, well, maybe, but we were never really sure. And I was like, definitely coming up NG. So yeah, so you're good. I mean, I guess you guys could do not really a live reveal, but give them a call in the future on another episode and just be like, what'd you think when we called you? But I get one of those every couple of times I update. There's somebody that gets a big surprise.
Oh, those would be great videos, great posts for people to see. Because 6 genes, wow. That's awesome. Cool. So we have from FX. Are you releasing your findings to the public like Dr. Seidel is?
Yeah, we will eventually. Probably in collaboration with either Dr. Seidel or Heather and Alan. In Canada, or maybe all 3 of us, if it's one of the complexes all 3 of us work on. In scientific literature, it's a very slow process. So we will have a whole ball python genome for both a male and a female that are wild caught animals. And so that will be a cool paper. So yeah, there's a lot of things that we have that are in process, but it's not a fast thing. I knew some grad students when I was a grad student that, they're publishing things that they did 5 years before that. So stuff moves slow. What were you going to say, Shawn?
The other side of that answer is stuff that we're not collaborating with other folks like Hannah and Heather. We also use a very large lab to do the higher end sequencing that helps us identify where some of these genes are. So the stuff that we've exclusively come up with through those channels, we actually have a contractual obligation with those folks that when they're published, that their name has to be on it. They have to be able to read it, and ultimately we have the say over all of the changes, but they are helping significantly. And one of the reasons we get a better price from them is we've agreed to kind of co-publish those when it comes time to do that.
So right now our main focus is getting to 30 to 40 ball python morphs, and then getting 3 or 4 more species going. And then we'll put a little bit more effort into publishing, but we just want to make the tests available right now. We're all, the 3 of us are much more reptile keepers than academics. And I do have some academic in me still. Shawn gives me crap for it sometimes, but.
Well, the business side of it is look, this is a business. We have to make sure everybody's paycheck clears. And the way we do that is we have the large enough breadth of test in a shortest amount of time as we can, so that ultimately we can all do this full time, right? I would like to do this 100% full time all the time and not have to have a day job anymore. Kayla too, maybe someday.
Oh yes.
We're not there yet, and that's not because everybody's not using us. It's the problem is really we don't have enough tests across enough species yet. So yeah, the publications will come, but that's once we can slow down a little bit on new R&D and focus more on the science and the publications. Yeah. Right now it's all R&D all the time. And specifically like this week, it's also, oh my God, we have so many samples we have to go through.
Baby sheds. everywhere. Y'all, if you didn't see the box Shawn held up earlier, just go look at it.
July, today's the last day of July, and I haven't looked at the numbers for today, but we're twice as busy as we were in our very first month, which was our best month ever. That was our
year.
So this year this month we've done twice what we did that first month. So, and the first month started here and kind of went down as people used us and, experienced the 6 tests that we had at that time. Right. And now that we're back up to 16 tests in baby season, it's curved. So the plan is working. We just need more time, and for you guys to keep throwing us your sheds, both for research and occasionally send us a paid test so we can feed our kids.
And sometimes we have to make a decision whether we're going to try to get the samples that are here done really fast, or we're going to try to make a big step forward on, 1 or 2 or 3 of the new tests. And, sometimes we can't do fast and get more tests. We have to choose one or the other. And
it's actually kind of a good way to segue right now, because you and I talked a little bit about this. We have one more run going in next week, and then I'm gone for 10 days.
Oh.
So there will be people here prepping sheds and getting them ready to go. But without me being here, they're not probably going to ship anywhere. So Ben is going to back a little bit off of processing tests and focus more on R&D and closing out the 3 that are coming up, and hopefully maybe getting another 2 or 3 lined up and ready for, as I call it, testing the test phase. So in about 10 days, we're going to shift gears just a little bit for a couple of weeks and focus more on R&D to try and close out. We would ideally like to have another 6 to 10 sealed up and before we go back to Tinley in October, which will be our 1-year anniversary as a business.
Oh, well, that'll be cool and good to know. We have from Sam Fisher. Do you still want the oddball species for research, like anacondas, African rock pythons, Florida pine snakes, etc.?
Yes, for sure. I would love to.
Yeah.
I have sent off a very broad sampling of different species, especially python species, because that's mostly what I've kept over the years, are various pythons. But yeah, we've got some cool stuff coming in other species, whether it's locality or purity or colorations, other morphs and wild animals that we see, like different phases or whatever. So that's all stuff that we want to work with. There's really not an end to the depth of nerdiness. I want to go that way. So send them on in, and thankfully they keep really well. If I can't work on it for the next year or 2, I'll still have them. The sheds I sent off 2 or 3 months ago, I've had for about 5 years. So
yeah, thank goodness they keep well.
Yes, it takes time, but I'll get to it. I'll keep them, and I will get to it.
Okay, so we have from Royal Canadian Reptiles. What sheds are you guys looking to collect next? I've been stockpiling sheds.
Awesome, thank you. So I would say Monsoon, like we talked about earlier, that's the one I want to send in within the next couple of weeks. Stranger's another one that I'd really like to have some more sheds coming in for.
Hurricane.
What's that?
Hurricane. We get quite a few inquiries for Hurricane.
Yeah. People ask about Hurricane, Trick, kind of in that complex or similar types of morphs. There's another one I was thinking of. But yeah, it's pretty much any test that would be helpful to you. Oh, paint. That was the other one I was thinking of. We've had several people ask about paint. I don't have very many of those, so more paint would be great. Really special. I don't have very many special. That would be another good one. So yeah, just ones that you have several of. If you have enough that it's a test that would be helpful to you, that means you have enough that you can send some sheds in to me.
Yeah. Let's see. We have also from Jesse Corte. With more genes becoming available, will the cost of the all gene panel increase?
So that's a great question, and we touched on it a little bit on a previous live, and get Shawn's thoughts too. Our last time we talked about it is, as it gets to over 25, especially when it gets to 30, what we'll probably do is break them out into smaller panels. So we'll have like a recessive panel, we'll have a yellow belly complex panel, we'll have, I don't know, 3 or 4 or 5 different panels that are like 5 to 10 mutations each. Because a lot of the time when people are sending in, there's definitely some people that are like, hey, I don't know for sure what this is, I just want to test it for everything. But a lot of the time there's 1 or 2 or maybe 3 different mutations, and they're like, I'd be interested to know about 2 or 3 others maybe. But you don't necessarily need 30. So I think that's what it'll eventually morph into, is we'll have 3 or 4 or 5 smaller panels, and then we will have a genome registry that will give you the opportunity to test everything, and any new tests that come out, you won't have to send another set in. So that's what we're working toward. Is that kind of what you're still thinking, Shawn?
Yeah. So besides the fact that it takes a minute to update 16, 20, 25 genes on a normal panel, where we're really going here is we'd like for people to start sending us sheds on panels where we run the genome registry, and then we have that information forever. We don't have to pull a shed and stuff it in a tube and add a bunch of reagents to it and cook it and sequence it and then analyze the data. We don't have to do any of that crap anymore once we sequence it the first time. Once it's sequenced, we have your animal's fingerprint, and we can pull test data out of that forever. So that's really where this is going. We would like ultimately for people to either purchase a single test, one of our smaller 5 to 10 gene panels, which will be some number underneath what our panel sells for now, and then the big panel would be the genome registry from there on out. You want a result for everything? Fine. Send us your shed, and we're going to give you the end results. And in the future, by the way, any new tests that come out for ball pythons, we will be able to pull your results from that original sample that you maybe sent us years in the past. So that's where we're headed. Right now, the panel is working well, but it's tedious, and you still have to send us another shed months down the road when you get when we get a new test that's released. But in the future, we hope to be able to avoid that. And that price will probably be almost exactly, if not even a little cheaper than what the panel price currently is today. So
It'll be awesome once that's in.
That's where we're headed.
Yeah.
We're doing the very best that we can to give you as much as we possibly can at the price we're offering, and to continually make that more efficient and more valuable to you as a keeper.
Yeah. So we're at the 57-minute mark here. So let's do the last 3 questions before we call it a night.
That sounds good.
Sounds good. So this one is from Stephen Crugito. I hope I pronounced that correctly. Do you know when you'll have other axanthic lines? I have a clutch now from 2 visual VPI parents with no visual axanthic babies. Whoa. I'm wondering if one of the parents is actually a different axanthic line.
Yeah, that sounds very likely.
Yeah.
So us
Poor guy.
personally, yeah, it sucks when that happens. That's not fun. That happened, back in the day when the axanthics first hit. There's people that had put some serious money out, and I think it was a podcast Mike Wilbanks was on where I heard him talking about back in the day when they were doing that. It was one of those keepers breeders that's been in the game for a long time, back when you're paying tens of thousands of dollars per animal. But yeah, you're not the first person to have that happen, that's for sure. So we'll have TSK axanthic hopefully within the month. Hopefully we'll have the data within a couple of weeks, and shouldn't be too difficult to turn that around where we'll have a test ready. MJ, the Canadian group, is working on, and any others, we'll probably need more sheds coming in, but yeah, we'll slowly be able to. But what we can tell you is if one of them tests positive for VPI and the other one doesn't, that we'll be able to tell you real quick.
That's true. Okay, so we have another one from FX. Can you guys please post on your webpage all of the genetic morphs available for ball pythons, as opposed to just going to Clutch or Morph Market?
Yeah, we can certainly do that.
Yeah.
I don't know how fast it will happen after baby season when things are a little less crazy. Any other suggestions that you all have, information that would be nice to have on our website as a reference? One thing I started doing was to kind of have like a 1 page, maybe like a 2-3 paragraph thing to give you kind of some of the nerdy details on each test, like who found the mutation, what kind of a mutation it is, that kind of stuff. So I've been making that for each of the morphs that we have tests for. But yeah, if there are other things that you all would find helpful, let us know, and we'll, once we're through baby season, right now it's crazy, but once we're through baby season, we're happy to do that.
Yeah. And lastly, from Leviathan Snakes, would you guys consider doing a Patreon where someone could support each month and get potential discounts or earn tests after however many months of membership?
Yeah, we've definitely talked about Patreon, and I haven't done that. I'm not very familiar with how those are set up and what types. I know that you usually have like 2 or 3 tiers or something like that. Yeah, maybe we'll have to talk offline with you if you have some ideas, but I'm certainly open to it.
I've thought about it. I mean, I'm always thinking about ways to help us with the R&D budget. The thing is like, we're already doing this weekly sort of, here's the deep dive into what we're doing. I don't want to make that a pay to play thing, right? So, what do we offer the Patreon members besides what we're already doing? That's sort of the trick. I mean, a free t-shirt wouldn't do it for me, right? So, I just don't know. I like doing these, and I only do them once a month when we do the lives. You guys do them every week, and it's a pretty deep dive every time, right? So, if you want to get the Scooby on which ones are coming out first, I guess we could just make that a Patreon only kind of thing. But it's still, I don't know. I've always liked the idea, but this works so well, and we get such good viewership from it. I don't know.
What would be a worthwhile reward as that's for someone for your Patreon tier?
Yeah, what would you guys want to do?
Yeah, I mean, I could see like a monthly call where just Patreon members are there, and you could be a part of knowing what progress we've made on specific tests, and have some input on what people are wanting the most next. But I don't know what else we could do that would be outside of this. We'll have to think about it. If you guys have ideas, put them in comments or message them to us. Certainly open to it. But yeah, I'm kind of the same. I just don't know for sure what to offer.
Yeah.
Let us know in the comments or send us a message. We'd love to know, and I'd be happy to help get that set up. So yeah. And we are at a little over the 1 hour mark now for our live stream. So
can I say one thing? Lee asked about the mass DG test results.
Yes.
As the guy's managing that, I'm getting a lot of sheds in. The first 100 or so sheds that I've got will be on a plate and sent to Ben probably 2 weeks from now. Results would be 2 to 3 weeks after that. And then however fast the rest of the sheds come in, every 96 well plate that I can fill. So that's 45 to 50 people's envelopes with 2 sheds in each envelope. As they come in, I'll fill those plates. We'll send them off, but it takes us a little longer for those because it's not just an order number and an animal ID. I'm putting all of y'all's information into the same spreadsheet that Ben and I use for the paid results, except it's got about 14 different columns of information along with photographs. So it takes a long time to populate the spreadsheet and then stuff the sheds. And it's not priority because it's a research project. It's not these customers paid. So it gets handled when, like this week, we've been so crushed with business that I haven't touched it all week. So, but I can tell you that in the next couple of weeks, Ben will get the first plate. So if you're one of the first 50 or so envelopes that I got in, you'll have results 2 to 3 weeks after that, probably. Because getting the results back is easy. It's prepping them on the spreadsheet the way we need to see them to make them worthy research prospects. That's the hard part, and it's super time consuming.
Yeah, because we really want to make it available for us for research purposes, but also to be able to show you all that picture. So we've got a genotype. Here's a picture. Here's 7 that are the same. We thought were the same. Here's how different their genotypes are. Or here's 7 that have the same genotype. Look how different. Or look how these are all more green than yellow. Or that's what we want to be able to do. And it just now you all are taking part in research, and research takes time.
Right. Definitely takes time. So you guys have an idea of the volume that we're talking about. We gave you all 3 days to register, and in that time frame, we got well over 300 registered DG folks to sign up. And I've already gotten 100 envelopes easily with those sheds. So that's 200 sheds right there.
Yeah.
And there's another 400 coming if everybody sends me 2 sheds. Probably more than that, actually. So it's a big project. The faster your sheds get here, the faster you'll get results. But we can't guarantee a time frame because they're going to get run when we've got the time to do it the right way.
And it's baby season. So, but yeah, thank you guys for your patience in that. And it's so important that you're part of the research now. So good stuff. Just a general, thank you guys for joining us today, and make sure you like, comment, subscribe. Thanks to our Instagram people too who joined in with Ben and got to gaze at his voluptuous beard today.
Getting full.
Yes, it is. Yeah, Ben and Shawn, do you guys have anything else before we call it a night?
See y'all in a month.
See y'all in a month. Yeah.
Yes.
I only do it every month, so I'll see y'all in a month.
Well, there you go.
Thanks for watching. Appreciate it.
Thanks, guys. We'll see you next time. And outro in 3, 2, 1. And ending broadcast. You guys don't see this part. Bye.